Hi, my name is Dr. Alice Lee, and in Lecture 10, we will be talking about prokaryotic cell movement. Most cells can move on their own, but they can do this in a variety of different ways. So before we even dive into the mechanisms of movement, let's think about why microbes would bother moving anyway. Like most living organisms, they're moving in response to some type of stimulus. We call this tactic or taxis response behavior. The stimulus can be food. They're looking for nutrients like some carbon or nitrogen source. Perhaps they're fleeing predators or they're catching their next meal. Some common stimuli that microbes move in response to are oxygen concentration. We call that aerotaxis. This is actually demonstrated in the image directly here to the right. You can see here an air bubble. And if aerobes, which are bacteria that utilize oxygen, are placed in this environment, they will move towards the air bubble. Conversely, if you put an anaerobe, one that doesn't utilize oxygen, it will disperse from the air bubble. Similar responses can be observed with light, or phototaxis, and also toward chemicals that we call chemotaxis. Shown here on the bottom image, there's a chemical that's attracting microbes, like glucose, and they are seen in this dark cloud around the mouth of this capillary tube where the glucose is present. On the adjacent image here, you can see a clearish zone around the mouth of this capillary tube because it's releasing a repellent compound such as carbolic acid. Chemotaxis is a process by which organisms migrate toward nutrients and favorable environments. However, they will also migrate away from toxins and unfavorable environments. In many species of bacteria, this occurs when extracellular signals are detected by transmembrane receptors, which are then relayed to flagella motors, which then control the cell's swimming behavior, either towards or away from the stimulus. Now, most bacterial species are motile and move either by swimming through liquid environments or by moving across the surface. Most will move using flagella, which are long filamentous appendages that propel the bacteria. There are several different arrangements of flagella that exist. The peritrichous arrangement, where the flagella are distributed all over the surface of the cell, such as in panel A here. The monotrichous arrangement, where you have a single flagellum at one pole, that's shown in panel B here. You also have lophotrichous and polar, meaning a tuft of flagella coming from one pole, such as here in panel C, and amphitrichous and polar, flagella that are on both poles of the cell, such as in panel D here. Now, bacterial cells are capable of various patterns of motility, the ability of an organism to move itself. When a bacterium moves in one direction for a length of time, the movement is called a run or a swim. Runs are interrupted by periodic abrupt random changes in direction that are called tumbles. Then a run might resume again. Tumbles are generally caused by the reversal of the flagellar rotation, such as shown here in panel A, when the flagella are bundled and the flagella motor is moving in a counterclockwise rotation, a run will occur. But when the motor goes clockwise, the flagella will fly apart and the cell will tumble or free fall. Some bacteria with polar flagella will have reversible motors that allow them to move forward if it turns counterclockwise and backwards if it turns clockwise. Some are not capable of that and may only have one rotation orientation, called unidirectional flagella, at which point the cell will have to stop, reorient itself, and then continue and change directions and then continue along on its run. A flagellum has three basic parts. The long outermost region, called the filament, which is made of individual flagellin proteins. The filament is attached to a slightly wider hook. The third part is the basal body, which anchors the flagellum to the cell wall and plasma membrane. The basal body contains a small central rod inserted into a series of rings. The L ring is embedded in the outer membrane and the lipopolysaccharide layer. The P ring is in the peptidoglycan layer. The MS and C rings are embedded in the cytoplasmic membrane. The MOT proteins here function as the flagellum motor, whereas the Fle proteins function as the motor switch. The flagellar motor rotates the filament to propel the cell through the medium. A proton turbine model has been proposed to explain the rotation of the flagellum. Protons that are flowing through the MOT proteins may exert forces on charges that are present in the C and MS rings, thereby spinning the rotor and therefore the attached filament. It can rotate at around 20,000 RPMs with an energy consumption of only about 10 to 16 watts and with an energy conversion efficiency close to 100%. Many bacteria have hair-like appendages that are shorter, straighter, and thinner than the flagella. Much of them are used for attachment and for the transfer of DNA rather than motility. The fimbriae are one such structure. They can occur at the poles, or they can be evenly distributed over the entire surface of the cell, such as in this image over here on the top left. They have a tendency to adhere to each other and to surfaces. As a result, they're often involved in forming biofilms. Fimbriae can also help bacteria to adhere to epithelial surfaces in the body, which then helps the pathogen to colonize the host and establish infection. So they play a role in pathogenesis. The pili are usually longer than the fimbriae, and they number fewer than the fimbriae with only 1 or 2 per cell. The pili are generally involved in DNA transfer. They play an important role in connecting 2 cells for exchange of genetic material. And they may also attach to host cells during infection. The pili are a bit different from the fimbriae in that they can be involved in a special type of motility called twitching motility. In twitching motility, a special type 4 pilus extends by the addition of subunits of pilin proteins, which makes contact with the surface, or maybe another cell, and then retraction of the pilin subunits are disassembled. This is called the grappling hook model of twitching and results in short, jerky, intermittent movements. Another type of motility associated with pili is gliding motility, a smooth gliding movement of myxobacteria. Gliding motility provides a means for microbes to travel in environments with very low water content, such as biofilms in soil. Finally, some bacteria use special gas vesicles, which contain pockets of gases for buoyancy and movement up and down the water column. For more information, read Chapter 4 in the Tortura text. This illustration here is by David Goodsell. It shows a cross-section across E. coli showing all macromolecules at 1,000,000 times magnification. The molecular complexity of this bacterial cell is emphasized in this image, which features a rather large flagellum and the motor proteins that power its rotation.